So, in this experiment, we are going to be investigating the effect of temperature on rate of reaction. So, we've looked at marble chips and acid and surface area. If you're going for investigating surface area, you're going to need a solid because that's what you need to break into smaller pieces. But when we investigate temperature, there are lots of different reactions that we can use, because we could use the marble chips and acid experiment again. So we could do that and warm the acid and then add it to the marble chips and collect the gas like we did last time. Or we can do the sodium thiosulfate and acid experiment that we did when we were looking at how we can measure rate of reaction. So we're going to be looking at the disappearing cross. Okay, so in this reaction we need some hydrochloric acid, which I have got in this measuring cylinder here. So I've got 25 centimetre cubes of that, and I have got 10 centimetre cubes of the sodium thiosulfate. I always tend to keep the sodium thiosulfate in the small measuring cylinder and the hydrochloric acid in the big measuring cylinder, so I don't get them mixed up, because if I then measure them the other way round, we could end up with a reaction starting in the measuring cylinders as it starts to react with the residue that's left in there. So we try and keep those separate. So when you're doing this yourself, that's quite an important tip to take home. So I'm going to do this at just 3 temperatures. I've got this water bath set at 60, this water bath set at 40, and this bowl of water is just room temperature water, so that we can investigate the 3 different ones. Okay. In order to bring our reactants to temperature, I'm going to put the reactants into 2 separate boiling tubes in the water bath and leave them until they get to the temperature that we need, and then once they're at temperature, put them into the conical flask, which will be stood on the cross, pour them in, start the timer, and as soon as I can't see the cross anymore, and I'll move the camera so that you can see that a little bit better, as soon as I can't see the cross anymore, we stop the timer and we record the time. So here we're recording how quickly our product is being produced, because it's the product that makes it go cloudy. We get a precipitate in our solution which obscures the cross. Okay, so let's get started. I'm going to put my acid in there, and I'm going to leave it in there to stand, and my sodium thiosulfate in there. Now, it's just me doing the experiment today, so that's not a problem. When we do it in class, there are lots of you using the water bath, so you do need to make sure you label your boiling tubes so that you collect your 2 and you don't end up putting 2 boiling tubes of acid in your conical flask, because you'll be waiting a long time for a precipitate to appear. Okay, so I'm going to quickly set up the rest of these and relocate the camera so that you can see the cross more clearly as we do the experiment.
Okay, so our reactants are now at the correct temperature. So we're going to start with our room temperature reactants, and at room temperature we are currently at 23 degrees. Okay, so I'm going to pour the acid into the flask here, ready, and as soon as I've put in the sodium thiosulfate, we're going to start the clock. So we put it in and start the timer, and we're going to time how long it takes for the cross to no longer be visible. So you should be able to see through the flask to the cross at the bottom, and this one is the longest wait that we will have. Okay, and while we wait, you can see the timer going up there, so we're already nearly at 30 seconds, and there doesn't appear to be much happening yet, but give it time. So in this experiment, the things that we've needed to keep the same are the volume of the hydrochloric acid and the concentration of the hydrochloric acid, and the volume of the sodium thiosulfate and its concentration. So each time we repeat this experiment, those are 2 of our control variables, and they're the most obvious ones, and in an exam those are the ones that you're probably going to be expected to record. I don't know if you can see that at the moment. it is slowly disappearing. So you should be able to see the cloudiness appearing through the flask. So we can see the cross is slowly being less visible than it was before. We're already at a minute and a quarter. The independent variable, the thing that we have chosen to change in this experiment, is the temperature of the reactants. So that's the thing we're choosing to change. Independent variable is the temperature. The thing that we're measuring in this particular reaction is our dependent variable in this reaction is the time it takes for the cross to disappear. In a different reaction, we might be able to say, well, it's the rate of reaction, which is true, but the thing we're actually measuring is the time it takes for the cross to disappear, all right? And so that's the thing that we would write as our dependent variable if we're asked about this particular experiment. Now, I can just about see the cross through the solution. Deciding when the endpoint is quite tricky. Has it disappeared? Are we happy with that? So this is one of the sources of error in this particular experiment, is deciding the end point, deciding when the cross is no longer visible and therefore we can stop the clock. That might depend on the individual, but it also can change from one person doing it every time being a little bit less clear one time than the next time. Okay, I'm going to say that that has disappeared now. Okay, so we're at 2 minutes and 42 seconds. So that took quite a long time. But determining the endpoint is one of the trickiest parts of this experiment. And it's the biggest source of error. So if you're asked about a source of error in a sodium thiosulfate hydrochloric acid experiment, whether you're looking at the effective temperature or concentration, determining the endpoint and when the cross is no longer visible is the key source of error in this particular experiment. Okay, so we're now going to repeat the experiment with our next temperature. So our next temperature is 40 degrees. So I'm going to clear this one out. We'll move on to the next one and then we'll move on to 60 after that. So in goes our acid. It's actually at 37 degrees. Let's reset the clock. Okay, in it goes. Start the timer. So we're at 37. So we're pretty close. And stop. Okay, so we're 1 minute and 4 seconds. So I will record that on the board and reset, ready for our 60 degrees temperature as well. Okay, so here we go. And now we are ready for number 3. So this is 60 degrees. Okay, so the acid is going in and we will reset the clock. Come on, button. There we go. Okay, so I'm going to add the sodium thiosulfate and start the clock. As you can see, this one is going cloudy quite a bit quicker. And you see the kind of the pretty swirly pattern that it's making. And it smells very much of eggs at the moment. Sulphur's got quite a strong smell, so we get that sulphury smell produced. And stop. Ooh. I think it was 35 when I tried to push stop the first time, wasn't it? Okay, so we've done all 3. I'll write those results on the board and then I will show you what they are. So bear with me a minute. Okay, so there's our final results. So you can see our temperatures that we did, and I think that when we did room temperature, our temperature was slightly cooler than when I did the first run. So you can see that there's quite a reasonable discrepancy between the two. But in the second one and the third one, the temperatures are the same. because we were using water baths to maintain the temperature of the solution. So water baths are the best thing to use when you want to keep a controlled temperature, okay, because they are thermostatically controlled, so it keeps the temperature nice and steady. So a thermostatically controlled water bath. Biology and chemistry, sometimes you might be asked about how we might maintain the temperature. Okay, so these are our results. So if you are recording these at home because you missed the experiment in school, these are the results that you will need. Okay, you'll notice that I've just written the numbers. I should really have just written the numbers for the temperature as well, because we write the units in the top, and we've got 2 repeats. So you would find the mean of those and then draw a graph. This would be a line graph because we have got 2 continuous variables. We've got the temperature and we have got time, both of which are continuous variables, so this would be a line graph. We don't really have enough points to draw a very good graph with this. We ideally want at least 5, and in the exam, if they want you to draw a graph, they will probably give you at least 5 of those so that you can draw a decent graph. So at this point, we would draw our conclusion, draw our graph, explain our conclusion. So why does the temperature affect the rate of reaction? The higher the temperature, the more energy the particles have, so the faster they're moving and the greater the chance they have of colliding with the activation energy, with enough energy for the reaction to happen. Okay, and then we evaluate actually how reliable is it, what are the possible sources of error, and as I said, the main source of error in this particular experiment is determining the end point. So we could make sure we've got the same person and make sure that we're fairly clear about the end point. You could also use a colorimeter or a ray of light and a light sensor, which would determine an exact number, and when you reach that number you would push stop on your timer, because it tells you how cloudy or clear the solution is. Okay, so that's it for this experiment. Hopefully it was useful, and I will do a second one for when we do our concentration and rate of reaction.